In this video, we will discuss about cell cycle measurement via flow cytometer, and the main component that we will measure is the DNA content of the cells, and with with availability with the signals, or we can say with the measurement of DNA content within the cells, we can identify the condition in which the uh, cells is presently. With the help of DNA content, we can identify the present stage of the cells in the cell cycle. So before going into the main topic, let us first quickly revise what does this cell cycle means and how DNA content is related to each cell cycle. Let us start with the G1 phase. In G1 phase, what happens is that cells is continues to grow. However, there is not much change in the DNA observed. So in G1 phase, we are expected to see ideally a sharp peak without too much widening of the peak, and because there is not much change in the DNA content. In G0 phase, what will happens is that in G0 phase is the intermediate condition in the G1 phase, in which cells obtain a senescent condition. So what happens in that condition is that cells inhibit to grow, whereas perform its necessary function that is essential for its survival. And after that, the G1 phase, G0 phase is not a normal in regular cell cycle condition. It is a special case that observed because of uh, external environment or changes, epigenetic changes within the cells. So in after G1 phase, it will cell is moved towards S phase, where it will start to reproduce its DNA, or starts to produce a copy of its DNA within the cells. And during the S phase, this whole process will carried out. So what happens is that in the start of this S phase, the the beginning of the copy of the DNA happens, and as soon as it's moved from at the end of the S phase, uh, it it reaches to a condition where it has almost two copy of DNA. After that, what happens after that S stage, it will comes into the G2 phase. In G2 phase also, cells will prepare itself for the mitosis. That is, cells will uh, uh, cells will separate all the organelles that are produced during the S phase, and equally divide itself into two daughter cells that has uh, that are exact replicate of each other in case of normal condition. So cell will prepare itself before dividing into the two cells in a phase that is called G2 phase. In M phase, what will happen is that cells will divide itself into two daughter cells, and after this after division of the cells in the daughter cells, each daughter cells will again start with the G1 phase. This is how the cell cycle functions. So in G1 phase, DNA remains almost constant. In S phase, there is a increase in the amount of DNA. In G2 phase, also DNA remains constant. In M phase, also DNA uh, remains constant. As soon as the after M phase, the DNA will significantly reduce, and as it reaches into the G1 phase. So this flow cytometers diagram is shown. Example of flow cytometer diagram is shown on the right side. So here we can see that the G1 phase, there is a very narrow peak because the change in the DNA is very less. And S phase is very broad peaks because there is a continuous increase in the amount of DNA. and less number of cells are in S phase and more number of cells are in G1 phase. From this figure we can uh, predict that most number of cells are in G1 phase that are used for this experiment. After this S phase, there is a G2 phase. In G2 phase, also there is not much increase in the DNA content. So it is this peak is also narrower than the S phase, and the intensity of peak is low. This indicates there's less number of cells are in G2 phase. So in this experiment, from this experiment, we can predict that cells that are used for this experiment are mostly in G1 phase, and some of them are in M1 phase also. This M1 phase might relate, or we can say might relate to some of the cells that are dead, because dead cells has very low amount of DNA, as DNA degrades with time, and this maximum number of cells are in G1 phase, some number of cells are in S phase, and whereas some number of cells are in G2 phase. So this is how we can make a prediction about cell cycles in the cells, make a prediction about cell cycles in the cell with the help of flow cytometer. The dye that can be used uh, can be either DAPI or propenium iodide is most commonly used in the for the measurement of cell cycle in flow cytometer. If you need to understand more about this cell cycle measurement via flow cytometer topic, you can go through the reference that has been provided in the description or references. That's all for this video. Thank you for your time.